I'd like to thank you for allowing me to talk today. And throughout the afternoon, you probably heard a lot of signature analysis is occurring, but along that process, we need some standardized way to perform this analysis or some kind of pipeline. So over the past year at UCSD, we've been developing this pipeline, and I'll go ahead and give a very simplistic overview of that today. But before I do that, I'm going to give a little bit more of a background, and just in case anyone is still unclear what a mutational signature is, we'll do a little bit more. So to do this, we're going to look at the lineage of a cancer cell. So from the point at which the egg is fertilized, we begin accumulating mutations. And now these might occur at very low levels throughout the lifespan of the cell. And at a certain point, other processes might start to act that introduce other types of mutations through childhood, adulthood, and eventually this might lead to some clonal expansion, eventually leading to a benign tumor. And these mutations will continue to accumulate, leading to an invasive cancer, a late invasive cancer requiring treatment, and what might eventually happen is you have a reoccurrence that's chemotherapy resistant. And so underlying these mutations are a set of processes. So hypothetically, we could have some process A that's occurring throughout the lifespan of the cell at very low levels. We could also hypothetically have another signature present that is occurring at only certain parts of the cell lineage, but at higher doses, and a third process that's only occurring at the end of the cell's life. But as a researcher, what we ultimately see is a final cancer genome. So we sequence the cells, and we get the accumulation of all the processes together. But ultimately, what we want to do, as everyone has described earlier, is try to associate or extract these signatures and then try to associate putative etiologies for each of these. So for example, process A might be some clock-like signature, while process B is attributed to UV damage, and process C might be due to platinum therapies. So ultimately, we start with our DNA in a very simplistic manner. We sequence, we align, and we call variants. But next, before we can perform signature analysis, we need a way to classify these mutations. So we have various ways we can do this, but ultimately what we want to do is create a matrix which represents our entire data set, where the rows represent the different contexts, and the columns represent the different samples in our data set. And now this ultimately allows us to look at mutational patterns that are representative of this data set. But to actually understand what's going on underneath, we need to perform an extraction. And this then allows us to perform the other downstream analysis steps. So as a whole, the packages that we've been developing encompass this portion of the pipeline, which we've labeled as Cig Profiler. So I'll go ahead and go into an overview of each of these tools, but we'll keep it fairly basic. So we have essentially 5 tools currently publicly available as GitHub repositories, as well as Python packages, and which made a lot of people happy as we converted the original MATLAB code to Python now for the extractor, and I've continued this same habit with the rest of our tools. And we have several other additional tools along the way, but they're not ready yet for public release. So the first tool in the pipeline is Cig Profiler Matrix Generator, which represents a standardized way for us to classify the different mutations. So ultimately, we take our somatic mutations after the variant calling step. We classify them. Here we're showing the 6 basic mutation types. We also include multiple different ways to classify single base substitutions. For instance, looking plus or minus 1 base before and after the mutation, plus or minus 2 bases, incorporating transcriptional strand bias information, and so forth. We also do this for double base substitutions and small insertions and deletions. And immediately, this allows us to look at mutational patterns and provides input for the extractor. So while currently there are other tools available that perform similar functions, we've shown that our tool is actually capable of exploiting all the different contexts and maximizing its functionality while providing plotting functions for most of the different contexts. This is currently in preprint, where we describe in detail how we classify each of the matrices, because as someone new to the field, it becomes quite difficult to actually understand how we classify transcriptional strand bias, or what does it actually mean to have plus or minus 2 base contexts, double base substitutions, small insertions and deletions, and so forth. And we also compare benchmarking to show that our tool is actually performing at a higher degree in efficiency than the other tools, while providing additional functionality. The next tool in our pipeline is Cig Profiler Plotting, and this allows us to visualize the mutational patterns in our sample, as well as the mutational signatures. So here's an example of a sample portrait for a breast cancer sample, where we have displayed all types of plotting available in the tool. So on the top left, you'll see, or on the whole left side actually, you'll see all the single base substitution plots. In the very top left, we have our 6 basic mutation types along with transcriptional strand bias. And then we've incorporated the 96 plot that we're all used to seeing with transcriptional strand bias. And now we've also incorporated a 1536 plot. So hopefully this will provide more insight and is easier to recognize new patterns when we start to move towards the 1536 classification. On the top right, we have our double base substitutions with the standard 78 classification system along with the transcriptional strand bias. On the bottom right, we have our small insertions and deletions again with transcriptional strand bias. So the next part of the pipeline is de novo extraction of signatures. And now this part of the pipeline also provides an automated selection for a solution. So essentially this tool will take in the mutational catalogs or the matrices generated by the matrix generator and will perform a de novo extraction of the signatures that are operating within these samples. It will also provide a weight, so how much each signature is acting per sample, along with a reconstruction error. And so it performs several iterations, increasing the number of extracted signatures, and ultimately chooses an optimal solution for you, but you also have the ability to go in and select other solutions or dive more deep into those. So to test how efficient or how well this tool characterizes a known data set, we took a simulated data set where we knew they had 6 ground truth signatures. We performed the extractions, hypothetically. Let's say we have 4 extracted signatures. And then we compare how these extracted signatures matched up with the ground truth. So to determine how well our tool is working, we looked at several different factors. One being the number of true positives that were reported, or those that were found in the extracted solution and also found in the ground truth. We looked at the number of false positives, or those that were extracted but aren't actually represented in the ground truth. And we looked at the number of false negatives, or those that were found in the ground truth but our tool is not able to extract. So we compared our tool with simulated samples and also Signature Analyzer, which is a tool developed by the Broad Institute. And so we can see in our first example is we took 1,000 samples with 39 ground truth signatures, and you can see that both tools were able to extract a similar number of signatures. However, Cig Profiler is capable of extracting more true positives while reporting less false positives and less false negatives. We repeated this and accumulated the results over about 18,000 samples with 16 different data sets. As you can see, both tools detect the same number of signatures, but again, Cig Profiler extracts more true positives and reports less false positives and less false negatives. So we're pretty confident that our tool is working to the best of its ability, but it's still being further optimized currently. A similar tool that we have in our collection is Cig Profiler Single Sample. So this allows you to take, if you have a small data set or a single sample, and attribute a known set of signatures to that sample. So for instance, if we had a single lung cancer genome, we could take a known set of signatures, such as the cosmic data set, and attribute what signatures are making up that sample. And so we have a small working group, as Sergey mentioned earlier, that's working on optimizing the attribution step. So this tool is still under development. So the last tool I'll talk about today is Cig Profiler Typography. And this represents the downstream portion of the analysis. So we really want to look at how these mutations and mutational signatures are behaving across the genomic landscape and across different cancer types. So for instance, we can look at nucleosome occupancy, or how these signatures are behaving around the presence or absence of nucleosomes. We can look at replication and transcriptional strand bias. So is there a preference for mutations falling on the leading or lagging strand, or on the transcribed or untranscribed strand in protein coding regions? And we can also look at replication timing. So are mutations more likely to occur in early or late replicating regions? So some preliminary analysis we did with the Mutographs data, the esophageal squamous cell carcinoma, is we took all the mutations attributed to signature 2 in the data sets, which in this case is about 100,000 mutations, and we calculated a nucleosome occupancy score at the point of the mutation, as well as 1,000 bases before and after. And what you can see is that there's an increase in nucleosome occupancy at the point of the mutation. However, if we go ahead and simulate this data, by chance, we actually expect there to be a higher nucleosome occupancy signal, suggesting that signature 2 is preferentially causing mutations that are in lower occupancy regions. Similarly, we can look at all the mutations attributed to signature 17B, and you can immediately see that there's an increase in nucleosome occupancy at the point of the mutation. And there's also now this periodic signal surrounding the mutation, suggesting that the mutations attributed with this signature are occurring in regions of clustered nucleosomes, especially when you compare it to the simulated data, which actually shows that there's a decrease in nucleosome occupancy expected by chance at the point of mutation. Similarly, this tool also allows us to look at replication strand bias, so whether or not mutations are occurring on the leading or lagging strand. So here we've taken all of the mutations in a set of breast cancer data set attributed to signature 2, and you can automatically see that there's an increase in C to T mutations on the lagging strand when compared with the simulated data. Similarly, we can perform a similar analysis with transcriptional strand bias with the same breast cancer data set, except these are all the mutations now attributed to signature 5. And you can see that there's an increase in T to C mutations occurring on the transcribed versus the untranscribed, which is different than what we'd expect by chance looking at the simulated data. And the final analysis that this tool provides is replication timing. So whether these mutations are occurring in early or late replicating regions. So here again, we've taken the breast cancer data set and all the mutations attributed to signature 5, and you can see that there are more mutations occurring in late versus early replicating regions. And when you compare this with the simulated data, we'd expect more of a uniform distribution. Now, if we compare this with the ESCC samples, we see a similar behavior. However, if we now look at signature 13 in breast cancer, we see a more uniform distribution with the mutations attributed to this signature. However, if we look at the ESCC cells, we see that there are more mutations occurring in late versus early replicating regions, suggesting that these signatures might be behaving differently across different tissue types and cancer types and the genomic landscape. So in conclusion, we've essentially developed a standardized pipeline that allows you to go from your variants all the way through to signature analysis. Ultimately, we're attempting to provide Wikipedia-style documentation that allows more ease of use, because it's very difficult for a new user to try to use bioinformatics tools. So the idea is we want to make these tools as usable as possible to the general public. Similarly, to do that, we're going to add a web integration, which allows more of a drag and drop style for smaller analysis and preliminary data. And then we'll also continue publishing technical reports similar to the matrix generator that's under review. And in future releases of the tools, we hope to start including structural arrangements and copy number changes, as well as start to include some epidemiological data along with the analysis. So I'd like to acknowledge the UCSD Alexandrov Lab and the members at the Duke Medical School and everyone at Sanger and IARC for helping with all of us in the development team debugging and being patient as we work through all the bugs. Thank you.